Hello students, let's study blood flagellates. So the diagnosis is Kala Azar and the causative agent is Leishmania dononi. Leishmania dononi, the morphological forms are four: amastigote, promastigote, epimastigote and tripomastigote. The life cycle, amastigote form is a diagnostic form found in man. Promastigote, infective stage to man, found in insect vector. So vector is female sandfly and the species is Phlebotomus argentipus. Transmission is bite of the sandfly. Life cycle. So man and other hosts acquire infection by the bite of female sandfly. Then regurgitation of promastigotes form GERD majority, directly discharged from proboscis minority. So promastigote form transform into skin macrophages. Then they convert into amastigote, multiply, rupture of parasitized cells. Amastigotes are carried in the circulation, invasion of other reticuloendothelial cells. Amastigotes are released into blood and body fluids. Sandfly takes up the amastigote while feeding on infected host. Amastigote form transformed into promastigote in the gut, reproduced by longitudinal fission. Then promastigotes migrate to foregut proboscis, and then again they are taken up by the into the man or other host by bite of the female cell. Pathogenicity of Leishmania dononi. So clinical forms: visceral leishmaniasis, also called as Kala Azar. In Hindi, it is black fever. Post Kala Azar, dermal leishmaniasis, and the cutaneous forms, cutaneous leishmaniasis. Hallmark of Kala Azar is pentad of fever, progressive weight loss, hepatosplenomegaly, pancytopenia, that is anemia, leukopenia, and thrombocytopenia, hypergammaglobulinemia. Clinically presented as lymphadenopathy, hyperpigmentation, pedal edema, and ascites, and pancytopenia. Lab diagnosis: specimen, splenic aspiration most sensitive, bone marrow aspiration most commonly preferred, lymph node aspiration, liver biopsy, peripheral blood smear in HIV infected people. So microscopy, GIMSA staining detects LD bodies, that is Leishmania dononi bodies. So they are macrophages filled with amastigote forms. Culture detects promastigotes, useful for species identification and drug sensitivity testing. The culture medium is NNN medium. Antibody detection in serum by ELISA, the ICT, then antigen detection, carbohydrate antigen in urine, latex elimination test, molecular method PCR, real-time PCR detecting specific kinetoplast mitochondrial DNA, Leishmanin test, Montegro test indicates good cell-mediated immunity, delayed hypersensitivity reaction, positive in all stages except active BL and diffuse CL. Animal inoculation, cold hamster is used. Non-specific test, hypergammaglobulinemia by Napier aldehyde test and Chopra's antimony test. Pancytopenia by complete blood count. Pentavalent antimonial, liposomal amphotericin B, miltefosine, paromomycin. Prevention, National Vector-Borne Disease Control Program, vaccine trial and control measures. So control measures are personal prophylaxis by using insect repellent or bed nets, control of canine or rodent reservoir. Phlebotomus does not fly high above the ground level, so it is nocturnally inhabited. So sleeping at top floors also can prevent transmission. Early treatment of all cases, mainly through ponotic VLN PK with post-Kala Azar disease cases. Third is the Leishmania dononi, infective form promastigotes, vector sandfly, transmission bite of the sandfly. Clinical manifestation: fever and hyperpigmentation, splenomegaly, lymphadenopathy, pedal edema and ascites, hematological abnormalities, pancytopenia, anemia, leukopenia, thrombocytopenia. Lab diagnosis: specimen blood, pancytopenia, and PBS LD bodies, that is amastigote form of the Leishmania dononi. Hello students, let's study trypanosoma causing sleeping sickness. So Trypanosoma brucei causes sleeping sickness. Trypanosoma brucei, African sleeping sickness in man. Testis fly, genus Glossina is the vector. And Trypanosoma cruzi, American Trypanosoma, Chagas disease, Riduvide bugs are the vector. Morphology: two forms, tripomastigote form and amastigote form. Life cycle: infective form, tripomastigote. So metacyclic trypomastigotes are transmitted to man by the bite of testis fly. Metacyclic trypomastigotes in punctured wound transform into long slender trypomastigote, multiply by binary fission, converted into short stumpy form, invade blood stream and migrate to various organs. Short stumpy forms are taken up by the testis fly during blood meal. Long slender form in midgut and hindgut of the mosquito, the testis fly. Then epimastigote in salivary gland, metacyclic trypomastigotes in salivary gland, and again by bite of testis fly they enter into the human. So pathogenicity, Trypanosoma brucei causes trypanosomal chancre, stage 1 disease without CNS involvement, asymptomatic, fever and lymphadenopathy, hematologic leukocytosis, thrombocytopenia and anemia. Stage 2 disease, CNS invasion, characteristic progressive daytime somnolence, hence called sleeping sickness with restlessness and insomnia at night, which may be related to increased prostaglandin D2 level in body. Behavioral and personality changes with apathy, confusion, fatigue, and loss of coordination may be observed. Lab diagnosis, useful samples, multiple blood samples collected during febrile period, chancre fluid, CSF, lymph node and bone marrow aspirate. Blood examination, trypomastigote forms are seen by serial blood sample examination performed by wet mount examination, peripheral blood smear, thick and thin smear, concentration methods, microhematocrit centrifugation if parasitemia is low. CSF examination necessary to confirm CNS invasion, detection of trypomastigotes in CSF, WBC count more than 20 cells in CSF with parasite detected in blood or lymph node aspirate. Then antibodies, antigen detection, molecular method that is PCR, culture inoculated into kiwi medium, animal inoculation, mice.
Hope you find this session useful for your understanding and for your exams. Thank you.